Hello and welcome to today's event. We're so excited to see you. And we're excited to bring together two different groups of people for our very first pitch event hosted here at Berkeley Lab. And this pitch event is specifically designed to generate conversations and connections for the Department of Energy's Visiting Faculty Program. Yes, we will refer to it as VFP, its acronym. And maybe Ingrid, you can even pop that information page in the chat while we're talking. So faculty members from colleges and universities across the United States and researchers from JGI, Kbase, and biosciences here at Berkeley Lab are coming together this morning for this event. Just so you know, during the event, faculty attendees will get to learn about their different research areas and the technologies and methods they use to do the work that they do. Then, after you hear from them, you will have a chance to jump into a new Zoom room to meet each other and discuss where your interests overlap. Our goal is for faculty who are really interested in making a connection with a Berkeley Lab researcher, or researchers can come together to put together a project that is mutually beneficial that you'd like to work on together during summer 2022 or beyond. So just to begin with, what is Berkeley Lab for folks that don't know, or who maybe have just done a quick Google search about the institution? So what is it that we do? Who are we? There is way too much information to share with you than I actually have time for in this quick intro, but this is a really cool place to work. We're part of a large network of US Department of Energy national labs across the country, and we are one of the places that really carries out work aligned with the mission of the US Department of Energy. So that mission is to ensure America's security and prosperity by addressing its energy, environmental, and nuclear challenges through transformative science and technology solutions. All of that is to say, many of our projects at Berkeley Lab have applications in energy and in environmental science and environmental applications, and you'll hear more about that in a few minutes. We have about 4,000 full-time staff. About 250 of these also hold faculty appointments at colleges and universities. And yes, we have those accolades. We've won 14 Nobel Prizes, have 88 members of the National Academies, 14 National Medal of Science awardees, and our researchers generate approximately 2,000 publications each year. But my favorite part about working at the lab is the fact that we have thousands of folks, including undergrads, grad students, post-baccalaureates, faculty, who all come to the lab to collaborate alongside scientists and researchers, including those you'll meet today. So our staff, Workforce Development and Education, who have partnered with our scientists to host this event, our team promotes equal access to scientific and technical careers for students from all backgrounds through educational programming. Most notoriously, we run internships at the lab year-round across all of our scientific divisions and operations divisions as well. And we host these programs for undergrads, post-baccalaureates, recent grads from a four-year college or university, graduate students, and faculty such as yourself. So just to give a little intro about what VFP is for those who are new to this program. It is, again, funded by the US Department of Energy, and this visiting faculty program really seeks to increase the research competitiveness of faculty members and their students at institutions that are historically underrepresented in the research community. And this is done in order to expand the workforce really vital to the Department of Energy's mission areas. As part of the program, selected faculty members at colleges and universities across the US will collaborate with the Department of Energy lab research staff on a project of mutual interest. And faculty members may invite up to 2 students, one of which may be a graduate student, to participate in the research project. To be eligible to participate, you must be a US citizen or lawful permanent resident at the time of applying. You must be a full-time faculty member at an accredited US degree granting institution of higher education, so community college, faculty are eligible to apply. And we have a list of eligible or non-eligible schools, and we can provide that for you. And you must be a full-time faculty member in an area of either physics, chemistry, biology, and we do non-medical, engineering, environmental sciences, geology or geosciences, math, material sciences, or computer or computational sciences. And we can provide all of the information for you about the program. In addition, after this 1 hour event, we will have a 30-minute optional Q&A session in case any of those nitty-gritty details you'd like to ask for further clarification or just want to talk to our staff about. And with that, I will pass it over to my colleague, David. And I will just say, if you have a burning question and you want to communicate via chat, that's absolutely fine. But I'm going to mute myself and pass it along. Thanks, David.
Thanks, Alale. It's wonderful to see a lot of familiar faces and names on the call today, and looking forward to engaging further. So I'll share a little bit of information about the biosciences area and the Joint Genome Institute before we segue directly into our pitch group. So, biosciences at the lab represents the largest investment by the US Department of Energy in any one institution. So it consists of nearly 1,000 staff members and affiliates, as you can see here, across different job categories and career stages and such. And the three divisions, environmental genomics and systems biology, molecular biophysics and integrated bioimaging, and biological systems and engineering. The names imply some of the skills, attributes, and capabilities that are offered therein. And we're fortunate to have the BSE, or Biological Systems and Engineering Division Director, Blake Simmons, here to do a pitch. And Blake will also touch on the capabilities that are subsumed under the umbrella of something we're calling the BioManufacturing. And of the many hats he wears, I appreciate Blake's title, Vice President of Deconstruction for the Joint Bioenergy Institute the most. So as you can see here, I think the biosciences best personifies the spirit of team science that really was engendered when Lawrence launched this laboratory back in 90 years ago. In fact, we just celebrated this year the 90th anniversary. So a little bit about the Joint Genome Institute. And you can see our lovely new building that we moved into in December 2019, and then many of us had to move out of that building and still haven't been back. So I'm really looking forward to having all of us eventually coming together, whether it's this summer or later in the year when we host our annual Genomics of Energy and Environment meeting in that on the hill above the Berkeley campus at Lawrence Berkeley National Lab. We cohabitate in that building, the Joint Genome Institute, with the DOE Systems Knowledge Base. And you'll hear from Ellen Dow shortly about what is entailed in Kbase, and also the National Microbiome Data Collaborative, or NMDC. So we were established, the Joint Genome Institute, about 25 years ago to accelerate the progress in the human genome project. And we're the first institute to complete our obligation therein and step back and said, now what? And there was not any dedicated institution worldwide that was looking at the genomics of our environment and organisms that play a role in weaning ourselves from our dependence on fossil fuels. So that is the DOE Joint Genome Institute's niche. And you can see at the bottom here, this kind of NASCAR assemblage of infrastructure and institutional partnerships. We're all Berkeley Lab staff members, and that means we're also University of California employees fundamentally. Our budget's approaching $80 million a year and 285 staff. So We serve as a user facility worldwide to the community interested in environmental and energy applications through mechanisms such as a community science program or working with other institutions like the Environmental Molecular Sciences Lab at Pacific Northwest National Lab for additional metabolomics and proteomics capabilities through the FICUS program, Facilities Integrating Collaborations for User Science. Our biggest customers at the JGI are the bioenergy research centers, of which Kbase is one of the 4. And we do have the latitude through Nigel Monsey, our director, to jumpstart or catalyze new initiatives with partners through other mechanisms, including the Director's Science Program. So underpinning all of our science programs and access to our resources is scientific expertise in plants and fungi and algae, metagenomes or microbiomes, microbes, and also the platforms that you'll hear about of DNA synthesis and metabolomics from Ian Blaby and Ben Bowen, respectively. So as you can see, our user community spans the world and a diversity of environments from aquatic, coastal, soil, agricultural fields, Arctic permafrost, and extreme environments like Yellowstone hot pools, from which discovery of new enzymes, as well as new pathways for natural products and such are emerging. So as you can see here, our user community is mostly academic, and these are individuals that have applied through the competitive process and had proposals ranked for their scientific merit and also their scale, large scale. But also we have a community of data users that have free access to our data resources through our portals, and you can see over some 10,000 people regularly access these data. So getting back here, let's hear from the scientists themselves. So without further ado, I'd like to introduce you to my colleague, Simon Rue, who later on will join in the breakout room with Matt Kellum to talk about opportunities within the metagenome program. So please, Simon, take it away.
Okay. Hopefully the slides work all right. Hello, everyone. So like David just said, my name is Simon Rue, and with my colleague, Matt Kellum, I will present basically the JGI metagenome program and what we do and what we could do with you if you know, anything that I mention seems like it could connect to your own interest and research. Broad overview of what kind of data we like to work with and what kind of data we generate for our users. We are, like the name say, we are the metagenome program, so we are dealing with metagenomes. For anyone not familiar with this concept, very briefly, that means you start from a sample. Here you have a lake, Yellowstone hot spring, like David was mentioning, some soil. From this sample, you extract all the nucleic acids, DNA or RNA, and then you perform shotgun sequencing. You sequence small pieces here and there of these nucleic acids. And then from this data, there is some magic happening where you can reconstruct from these small pieces the genomes of the microbes and viruses that were here in your original environment. And that gets you a window into the microbial community and the microbial processes going on in these samples. So again, metagenome program, we are mostly dealing with metagenomes. We also like to do metatranscriptomes, which is basically the same thing, but targeting the RNA fraction, so targeting the activity more than, you know, more the who is doing what rather than who is there, basically. And we very much like to also have this metagenomes and metatranscriptomes part of a much broader set of data. We like this concept of index, which is a fancy way of saying, you know, trying to get as many information as we can from as many angles as we can on the same sample, basically. And at the end of the day, what we do the most, you know, on a day-to-day basis is this genome result analysis of microbes and viruses in the environment. Okay, so that's kind of the kind of data we like to work with. What are our main research questions, or our main kind of 5 and 10 years ideas for how to move forward. We are very interested into developing visual because we have this large-scale data that are very hard to explore if you don't have the right tools to, kind of dig through it and dive in it. So we are interested into this kind of technical aspect. We are interested into pushing the boundaries of which genomes you can get from metagenomes. The short genomes are relatively easy to get. The much larger and complex genomes, like microbial eukaryotes, are right now very, very difficult to get. And so we are interested into exploring kind of how do we get there. In terms of viruses, we like to look at which genes are encoded by these viruses. Viruses are really kind of the underexplored part of this microbiome. So a lot of novelty to discover there, a lot of new biology. We also are interested into linking these viruses to hosts and starting to reconstruct kind of these more complex phage host networks and these microbe interaction networks in a broader sense. And then we are broadly interested into anything that will help us connect the genomes we can reconstruct to actual metabolism and activity. And that's, for instance, more targeted assay like stable isotope probing. Okay, giving you one research product. So in a paper that we published recently, this is a work led by Stephen Neyfach and Emily Elofadrosh, who is the lead of the metagenome program. Emily is. And this was a massive paper where we actually analyzed more than 50,000 of these genomes reconstructed from metagenomes. And you can see on the tree here, we basically found a lot of different microbes. On the tree on the right, anything in black was known before. Anything is green is something new we kind of bring in. So you can see how much diversity and how much novelty we can find through metagenomics. Thousands and thousands of new viruses and their hosts, thousands upon thousands of new biosynthetic gene clusters, basically a lot of things still to discover and to survey in the environment was the bottom line of this kind of large-scale study. And I kind of selected this paper also to give you a sense of the type of scale at which we like to work at the metagenome program. We do like this kind of big data, thousands of genomes, and trying to sort through them. Some current projects, again, to give you a sense of the flavor of what we are interested in. On the left, this is kind of this first bullet point, defining boundaries between metagenomes using machine learning. It's basically this idea, and that's a project led by Matt. If you have a metagenome and a bunch of sequence, can you guess which type of environment it came from? And so that's kind of using machine learning, trying to say, okay, if you give me a sample and I'm blind to like exactly where it was taken from, can I tell you if it's a lake, if it's a soil, if it's, how granular can I get? So can I tell you actually which type of soil it is? Can I tell you that it's an agricultural soil? Things like this. Like I mentioned already, we are interested into pushing the boundaries of kind of the size and the complexity of the genomes we can get from metagenomes. And we're also interested into this, all of the eco-evolution question, basically. And for viruses, which are more of my specialty, this global diversity of all viruses and all the capsids that exist alongside, which give you a window into very ancient evolution of viruses, is also of interest to us. And I think that's pretty much the end of my 3 minutes. So again, to recap, we are the metagenome program. We have this kind of two research branches. It's environmental genomics, which is microbial ecology in general, and this viral genomics, which is more specifically focused on viruses and phages. And this is pretty much all of us right now. If you have any idea or any project that you will be excited about and where you think metagenomes could contribute, please, either join us in the next Zoom session or send us an email. You have our email addresses up there. Matt is here, I'm here, and that's pretty much it.
Simon Roux, everybody, going viral. Lale will put in the chat the link to the metagenomes breakout session for later. Next up, we have Ellen Dow from the DOE Systems Biology Knowledge Base, better known as Kbase. So take it away, Ellen, thanks.
Thank you, David. Okay, so hi folks, again, my name is Ellen Dow and I am a part of the Kbase user engagement team as well as the lead on the Kbase educators program. I am going to briefly cover how Kbase can enable your collaboration with the JGI. So what is Kbase? The Department of Energy Systems Biology Knowledge Base is a free open access software and data science platform that enables researchers to upload their own data and analyze it alongside collaborator data and within the context of public data. So this single platform can run various biological workflows, including metagenomic analyses and systems modeling for microbial communities, fungi, and plants. The narrative platform itself uses a graphical user interface that is built on a Jupyter Notebook instance, and this serves as a virtual bioinformatics lab notebook. So these workflows are created within the narrative by adding your research data to analyze using many tools that are typically only available on command line, but within the system, they are wrapped as apps for the Kbase user interface by our team or community developers. And so these workflows can also be complemented by text or images using Markdown cells. And these narratives can also be shared with and reproduced by your collaborators. And using the organizations within Kbase makes it really easy to share these narratives and data with your entire team. Kbase enables continued and collaborative research with the JGI. You can continue to analyze your data in Kbase, including those thousands of genomes that Simon mentioned earlier from the Genomes from Earth Microbiomes project. And you can use the Kbase's computational power with access from anywhere, alongside sharing data and analyses with your collaborators, in addition with students for projects and courses. Narratives in Kbase can be made static, which enables Kbase users to request a DOI for publication. And we also support educators. So through our educators program, we have been working with groups of college and high school instructors who have developed workflows across a variety of concepts, alongside best practices and supporting resources that can help adapt these narratives to your course curriculum and your class needs. We also have this great community of educators who work together and support one another. All of these resources are available within the Kbase Educators org, and really exciting. Last week, we just had our first publication from the project released, so you can check that on Frontiers. I have a bunch of these resources that are available for you linked within this slide presentation, but please come and connect with us during the breakouts. Thank you.
Fantastic, Ellen. Thank you so much. So the Joint Genome Institute is quite famous for generating sequence or reading DNA. In fact, we've achieved our second petabase of sequence just recently in the last month or so. What you may not know is that we also synthesize millions of bases and design and provide constructs. And that's what my colleague Ian Blaby will talk to you about now, the DNA synthesis platform. So please, Ian, take it away.
All right, thank you, David. Hopefully everybody can see my slides. Let me go full screen here. Okay, let me know if you can't see these, but hopefully slides are on the screen right now. So as David said, my name is Ian Blaby. I'm the platform lead for DNA synthesis for synthetic biology at the Joint Genome Institute. And over the next couple of minutes, I'll just give a bit of a brief overview of the capabilities that we offer before diving into one particular technology. So the group's mission is to harness the power of DNA synthesis, strain engineering, and biosystems design for DOE mission relevant discovery and applications. And we have four primary themes of focus as indicated here. So firstly, high throughput functional genomics, genomes to structure and function, which embodies crystallography and determining protein crystal structures, plant microbe interactions, and bioenergy research, of course. So really, in the interest of time, I can't unfortunately drill into detail on each of these, but to give you some kind of flavor as to what we do as a group and the capabilities that we offer, we comprise 12 team members split between software developers and computational scientists and molecular biologists. And we cover everything from high throughput DNA assemblies, whether this is single gene inserts into vectors, through to very large inserts, 100 KB regions, 100 kilobase pathways. We do libraries, whether it's Golden Gate libraries with tens of thousands of permutations for screening and optimizing production, as well as a more recent offering that we've just started offering of strain engineering. And all of these capabilities we perform at both the bench at a molecular level to deliver DNA constructs or engineered strains, as well as building our own biodesign and biocad and data analysis and visualization softwares. So please feel free to chat to me about any research topic you feel may be relevant to what we do, but I want to home in on one specific capability centering around high throughput functional genomics, focusing really on CRISPR technologies for interrogating gene function at a genome scale. And so in this, we use our own in-house custom-built gRNA secret software to design guide RNAs that target either a small handful of targets for very focused experiments, or we can go to full genome scale. We will produce the pools, the libraries, using the most appropriate CRISPR technology for the question at hand. We'll sequence the library to determine the representation of each variant in the pool and make sure that you're getting a fully representative population. The operator then transforms to their specific organism of interest and invokes either some sort of genetic with a mutant or environmental perturbations. So for example, elevated temperature. After a couple of generations of culturing, we will then resequence that cell pellet and determine quantitatively the influence of each gene's relevance or influence on as a consequence of that experimental condition. So that was an extremely fast and high-level overview, but please feel free to join me and with any questions in the breakout rooms to chat more about any potential interfaces between what we do and what we can offer and how we can work together. And with that, thank you.
Thank you, Ian. Great stuff. In complement to JGI's capabilities in generating DNA sequence and reading the sequence, analyzing the sequence, and designing and writing the sequence, we also have available assets in metabolomics, and that's what Ben Bowen is going to talk to us about now. So take it away, Ben.
Hey, everybody. So this talk is jointly between Catherine Louis, the Metabolomics platform lead, and myself, a Metabolomics program scientist. And we're going to tell you about what you can do in an integrated way with small molecule information like nucleic acids, amino acids, organic acids, along with the genomic information. So, many questions can be answered when you combine this new dimension of information about metabolomics and concert with other tools. So if you have some soil, for example, you might wonder, when are metabolites being produced and consumed in response to some external perturbation, or which metabolites are produced by which organisms, who's eating what, who's producing what. And this information can yield to hypotheses about various food webs and interactions between microbes and plants and the soil. Sometimes it's just a very general question. What metabolites are there? Maybe new metabolites that we don't know the biochemistry about yet, secondary metabolites, for example. How are the metabolites produced and consumed? And that links back to the genes. Which enzymes are responsible for making metabolites? And then kind of lastly, spatially, where are these metabolites located? Are they near the root tip? Are they deep in the soil? How deep? What's deep in the soil? So anyway, these questions are best answered with metabolomics in partnership with the other programs at the JGI. So for example, at JGI, we have the most diverse metabolomics sets of experiments that one could ever imagine. Everything from Yellowstone hot springs, as David mentioned earlier, to biotechnology strains and single gene mutations. The projects that are integrated with other data types like transcriptomics and genomics. A lot of projects have to do with soil metabolites, and many also have to do with secondary metabolites. Anyway, it's across the board. We have diverse expertise with a large, highly diverse sets of experiments, in comparison to a medical metabolomics lab that only looks at human tissues. So this is kind of a neat aspect of JGI's experience with how to process these types of samples. If you're having a collaborator that wants to compare eucalyptus to pine to fungi to soil, that's a different sample processing protocol than let's look at human blood across 100 individuals. So as part of this VFP program, you would engage the full protocol. And that begins with sample extraction, where you learn how to extract your samples. And that's not as easy as it may sound. And we use solvents to pull metabolites out. And we we take those metabolites and we run them through what's called liquid chromatography with tandem mass spectrometry. And that generates huge amounts of highly complex data where you see signals associated with the metabolites in your samples. We have two protocols, one for targeted and one for untargeted. In this case, it's a poplar leaf that's inoculated with a fungi versus a poplar leaf that's not inoculated with a fungi. And the targeted analysis yields information about what metabolites are changing. And we can then make that analysis a much less targeted and unbiased and look at all the signals in your sample. And these might be from newly uncharacterized metabolites. We do that with a service called GNPS, and that's called molecular networking, where we link metabolite signals together if they're similar, and we look at how they change across your samples. And then lastly, we also try to hunt back into your sample and find the gene that might be associated with your given metabolite signals. And this service is called Magi. So by working with us, you'd engage this whole pipeline. Likely on-site would be limited to sample extraction. And then the rest of this would be data analysis exercises. And thanks. I hope to see you in the breakout room.
Thanks, Ben. I encourage you to join Ben and Catherine in their breakout room, find out about the opportunities in our Metabolomics platform. So last, but certainly not least, the man who's got his fingers on the pulse of all things biosciences, I now give you Blake Simmons.
Thanks, David. I don't know about any of that, but it sounds really great. And I wish I could have my fingers on the tap of everything in biosciences. But what I am here to talk about is potential interactions between the visiting faculty program and the BioManufacturing at Lawrence Berkeley National Lab. It is basically, as David alluded to, an integrated bioengineering effort that is really aimed at some mission needs enabled by core science and technology. It goes all the way from your idea or what you're working on in your project or what your problem statement is. And then by harnessing all the resources that you've heard about in the JGI and some the Joint Bioenergy Institute, there's Agile Biofoundry, and our scale-up unit, the Advanced Biofuels and Bioproducts Process Development Unit, you really have the opportunity, should you come and choose to work in this endeavor, to go from an idea to something that is demonstrable and impactful. And you can do so by stepping into and harnessing any of the capabilities here at any step of the way. It is really about bringing together all these capabilities to accelerate the discovery and development of bio-based products while diminishing risk for you instead of industry. This is for VFP. We combine several different platforms that we have at Berkeley Lab in terms of the multiple omics technologies, the automated workflows, predictive scale up, and some of the large scale fermentation recovery capabilities. And then those all can be fed into a high performance computing framework, as well as bioinformatics and modeling infrastructure. So this is encompassing all the things that you've already heard about, but aligning it in an integrated way to go after a specific mission area or a specific big topic that you are interested in. And so it could be anywhere from looking at economics and what it means from the target that you have, all the way through to feedstock engineering, to strain development, to fermentation development, and downstream processing. And again, you don't have to use all of this. You can plug and play anywhere along the way. So let's say you have a strain that you want of organisms that you're working on, and you want to come in and harness some of the capabilities of JGI, JBA, and Agile Biofoundry to increase tighter yield and productivity. You could do that, and then you can end the project by demonstrating it at scales that matter and generating enough of a sample so that you can actually do something applicable or mission-oriented with it, such as materials testing or proving that it can be purified in an economical way. Just a couple of examples of what you could do and what we're currently working on. At the Joint Bioenergy Institute, we're really focused on the sustainable aviation enterprise. So we'd be very interested in working with folks in the area of fuels, especially those that have high energy density molecules that could extend range and decrease greenhouse gas emissions. Looking at an interior cabin material, we're looking a lot at fire retardant polymers and different renewable fabrics. And then we're also looking at renewable carbon fiber and carbon composites and some biodegradable polymers that could go in and create some long-lived solutions. We're also looking at some mission-oriented areas such as wildfire risk, mitigation activities. So how could you take forestry thinnings and residues and convert them into biofuels, biopower, and biochar? I noticed there's some folks from New Orleans. I went to grad school at Tulane. One of the original embodiments of this was to develop a unit that could go into disaster hit areas by hurricanes or earthquakes and go in there and harvest the debris material and generate power to enable the recovery efforts. We're also looking at integrating biology with mobility. How could you take the biofoundry approach in engineering organisms for optimized hosts and pathways and then putting them on a mobile platform so you can do the same thing, but instead of limiting your outputs, you could tailor them into biomaterials, biopower, biofuels. So with that, I'd be delighted to talk more in the chat rooms. I kind of rushed things because I know we are pressed for time. And with that, I will stop. Thank you very much.
That was perfect. I just wanted to let everyone know that all of the Zoom links are organized for you on page 2 of your agenda. I've just put the link in the chat. Both Berkeley Lab staff and faculty attendees can feel free to hop into those. Our staff will stay in this main room in case anyone has any issues or questions, and then we'll run our Q&A session at the end, but feel free to go over on time as needed. This is the end of our formal presentation, and I hope you enjoy your networking. See you all soon.